previously talked about the effects of physical agents or factors on bacterial growth. So now we're going to talk about chemical agents. We're talking about any type of chemicals that can inhibit or prevent the growth of microorganisms. Couple terms to keep in mind, and yes, we're very picky in microbiology about our definitions. An antiseptic is something that's going to be used on skin. So it's being used on living tissue. Disinfectants are used on inanimate objects. So you can use an antiseptic. It's often used before surgery to clean an area of the skin. A disinfectant you would never use on your skin. You can wipe a tabletop down with a disinfectant. So our terminology is picky. So how are we going to test them? What you do is, first you have your Petri dish. Usually we have like a nutrient agar or some standard based medium that just about anything will grow on. So you have that Petri dish already made. It's obviously sterile. The organism you're wanting to test, what you do, now in our lab we're going to be working with a pure culture, but what you're going to do is take that, we usually use a cotton tipped applicator to inoculate the Petri dish. So usually your culture's in a liquid broth. You take the cotton tipped applicator, dip it in using aseptic technique, and then with the end of that cotton tipped that now has the bacteria on it, you're going to rub that across the surface of the agar on the Petri dish. This is creating a solid mat of growth there that we call a lawn. So you want to create a bacterial lawn. Properly dispose of the cotton tipped applicator after you've inoculated the plate. Then what you're going to do is you take little paper discs, and honestly in our lab what we often use are the little paper circle tabs from when you use a hole puncher. We save those tabs, sterilize them. You take so that's about the size we're talking about. You take that paper, you dip it into whatever chemical agent you're wanting to test. It might be Listerine, it might be Scope, it might be bleach, whatever the items you're wanting to test. You dip the paper in it, and then you carefully place that on top of the on top of the Petri dish. You spread them out evenly, so there's lots of space between each of the discs. You can usually fit 4 to 5. I usually put 5 on a plate. And then you're going to incubate that Petri dish. After 24 hours, maybe 48, but usually 24 hours is sufficient. You're going to pull the plate and you're going to look at it. Around the disc, if the chemical is able to inhibit growth, you will see a zone of clearing around it where there was no growth, because the chemical diffused out of that paper disc into the media and it prevented the growth of the bacteria. This clearing zone is known as the zone of inhibition. The idea is the greater the size of zone of inhibition, and sometimes you actually take a ruler and we'll measure it. So the greater that zone of inhibition, the more effective that particular chemical agent was in inhibiting the growth. So that's one way that you can test. Metals are known to often prevent microbial growth, and so you can also test this if you wish. It tends to interfere with the enzyme activity, and if the enzymes can't function properly, chemical reactions aren't going to occur, and so then the organism is not going to grow. Usually a very low concentration is effective for that. Sometimes what you can do, if you wish to test this, we don't tend to do it in the lab, but I've seen it where it's done, is once again, you take a Petri dish, you make a bacterial lawn. Instead of putting the paper disc, you can put a little piece of metal. Sometimes people will use coins and put like a dime there or something to see the effect. In terms of some natural chemicals that inhibit microbial growth, in tears and also a little bit in saliva. There's an enzyme called lysozyme. This does contain antimicrobial activity to it. If you think about it, it makes sense. The tears that are constantly flushing and lubricating the eyes, this helps to prevent infection of the eye itself, plus also prevents any bacteria from moving through the pupil and being able to access the interior part of the body by going through the brain. Some other natural chemicals, things like garlic, horseradish, onions, those all have antimicrobial activity as well. Certain fruit juices, because they're acidic, bacteria don't like a lower pH. They don't like acid environments, so that will naturally inhibit. And alcohol. Alcohol will actually kill a lot of microorganisms. That's why you see a lot of things like hand sanitizers contain alcohol in it. That's often how they work. This picture shows some plates after they have been incubated, where they're this first plate over here, you can see this nice kind of cloudy looking color. That is because this is a lawn of bacteria. This is solid bacteria growing on here. And you can see how the paper disc were replaced. None of these seem to be inhibiting the growth, but it shows overall the plate on how it looks. On this particular plate right here, this disc, you can see this clearing circle around. That is the zone of inhibition. And over here on this disc, there's a wider, much larger zone. So this disc over here, the chemical on here is much more effective than this first one over here. So that just gives you an idea of what the plates look like, how you can test different chemicals. If you think that you've come up with a new disinfectant, this is a way of testing. Well, is it really effective or not? Is it better? Or maybe it's so-so. But what are the side effects of the chemical to the human body that you're trying to come up with? So if you're wanting to take several different chemicals and test them, this is a quick, easy way of doing so.